Okay, so I'm really glad to speak with you today. It's a really a good format, and I will share some of the results of our research of the last few years. And I want to first acknowledge my co-authors here, the students Dan Rousseau, Julie Malas, and Jennifer Yak, and of course the PI on this adventure is Darcy Mayer-Dunbar. And so the first thing we have to look at is essentially the range of habitat on Earth that are high pressure. And we can fit in that environment, which is marked on that plot of pressure versus temperature by this red line, dotted line. We can fit in there several of the world's ocean. And of course, you wonder why this is, if you wonder why this pressure scale is exaggerated, it's to leave room for Titan. And because I'm part of the Hydrocarbon World Astrobiology Institute, so Titan and beyond, what I present is mostly relevant to Titan because we talk about experiments done at 150 megapascal, which is the equivalent of what you could find just below the ice cover, the ice crust on Titan. So these are the range of possible habitat. Of course, we know that we have I6 in this region, so there might not be an habitat in the ocean itself. So a very schematic view of what we are working on is and working with. So we try to understand the effect of pressure, and in that slide, in the top part, in the A part of the figure, you can see I drew very schematically something that illustrates what membrane do when they are submitted to stress of pressure on the left, top left, getting into gel phase, or high temperature on the top right, where the membrane goes into fluid phase. And so the membrane tries to stay in away from fluid phase and from gel phase by changing the constituent, its constituent. So you have examples here of what Archaea does at high temperature with tetraether lipids across membrane that help stabilize the system versus lower pressure system and cold, where they use shorter tendency to increase the concentration of C20 hydrocarbons, so half membrane spanning lipids, and so diether compounds. So for bacteria, there's many differences from using, again, ether-bound cross-membrane diabolic acid to stabilize membrane, from including unsaturation to create disorder in the membrane when you are in the gel phase and allow this membrane to function normally. So it's a game where not only you change the lipid constituents, and I will show that in more detail in an example, but also where you are susceptible to change the entire lipid, so the intact lipid present in the membrane, so the lipid with its polar head. So the lower part of the figure is the equivalent, very schematic view of what happens with different pH in Archaea on the bacteria, and again, the same type of response for different reasons. So what I will present in this here we'll talk about intact lipids. So lipids of the type you have on the left here. So we have an intact lipid with a substituent R, and these substituents can be the various one noted here: ethanolamine, choline, serine, glycerol, and others we are not concerned about today, like inositol, sugars, for example. And we have the other parts, so that constitutes the head, the polar head of the molecule, and we have the hydrophobic part of the molecule with the two fatty acid substituent here that can have a different conformation, as noted on the previous slide. So we will meet also cardiolipin. It's also a phospholipid, but it has 4 legs. So it has a head that is wider, but has 4 substituents on it. And ceramide, which is not a phospholipid, no phosphate in there, where you have an amide that link one of the substituent to what was originally here a glycerol. So what is important to note is the difference between these heads make the property of the molecule change, and its ability to work with the membrane change. And so we will be interested in both the changes in these. So phosphatidyl ethanolamine will be summarized as PE, phosphatidyl serine as PS, and PG will be phosphatidyl glycerol. And as we and I'll show you here the results we obtained with Staphylococcus. So the difference between the distribution of these lipids between atmospheric pressure and 150 megapascal, and you can see that we have a very vast change in the distribution of these intact lipids. So this phosphatidyl glycerol, which was representing more than half of the constituents in the control, are not, well, they are present here between the PE and the PS. They are very small amount. They essentially disappeared and left room for PEs and ceramide to occupy the entire space. So this is happened also in our third experiment. So Staphylococcus wanneri experiment 3, where we had other constituents. So we had no PGs in that control. And after experiment, the system was dominated largely by ceramide, showing that there was a very fundamental change in the lipid associated with the membrane, so in the intact lipid distribution. So it was not always the case. So we, when we work with Shewanella oneidiensis, and I put 3 experiments next to each other, with the top representing the controls and the bottom representing the experiments at 150 megapascal for 15 minutes. And we see we have essentially the same constituents, and it seems that what is present in the control is really constraining what we find in the experiment. There are little changes, but no architectural changes of the sort as we have seen for the gram-positive bacteria, the cocci I just showed you before. So are they growing, those experiments? Are we dealing with growing organisms? So this is a count in the initial culture, and this represents the cell count right after the experiment, and the cell counts in days post-experiment, so 1 day and here 7 days. And there's really a clear trend showing that effectively there was growth during experiment, and obviously survival after the experiment. So there was growth. We have some limitation in the fact that some of this early growth could have been occurring in a very short amount of time between retrieval from the experiment and stabilization of the culture. But we try to really minimize that amount of time to less than a few minutes. So the transcriptome data also indicate growth and changes. And you have here an MA plot, a so-called MA plot, that indicates, look at the RNA sequence and essentially put in color in brown the transcripts that are significantly upregulated, and in green those which are downregulated. So also indication that things is happening not only on the lipid level but also at the RNA level, so in the transcriptome. And so when we look at the lipid for Shewanella and adiensis, for example, we can look at the difference between prior and the control and the experiment. And this is percent of PE, so that these are all our phosphatidyl ethanolamine, and we have on the x-axis all the various species of substituents. So some are short chain with 12 carbon or 14 carbon. You have longer chain ones with 18 carbon and 18 carbon, and then the number indicates the number of unsaturation, and every of the number which is odd, like for this one, the 15-15, which is largely upregulated, these compounds are actually branched. It's a C14 branched, and you will see that we suspect that this compound in part formed from alteration of other intact lipids by in situ enzymatic reaction in the membrane. So, for example, this 15-0-14-1, so these two legs, one with the C15 and one with the C14 with one unsaturation, could use this unsaturation to form 15-15, so put a branch, for example, here, and so we can have then an intact lipid with two branched substituents. So there are clear upregulated molecules, and I could show you a graph for each of these phosphatidyl species, the PGs, the PEs, the PS, and it would be absolutely boring. The idea is that you see that we have differential expression of some of these lipids the same way we have differential expression of the genomes with the transcriptome data. So what this is experiment 1, and experiment 2 is this 15-15 is a little bit less abundant, but the same compounds essentially are upregulated. And of course, the middle experiment had a rather different signal that we cannot completely explain at this point. But when we consider the average of these compounds, for example, within the PE, if we look at the saturated compounds, they are actually upregulated. The mono unsaturated, in contrast to what we were thinking when we started that project, these are downregulated. And you see, quantitatively, they seem to be, these are relatively, they are significant numbers, and they are close to one another. the diunsaturated and pentaunsaturated are essentially not changing and very variable for the diunsaturated. So if we look at the same lipids, but we divide them into branch short chain versus long chain, so less than C18 and more, we see we have first, there's very little change in the long chain with relatively changing data from experiment to experiment, but essentially low abundance in the change of the long chain. And in contrast, we see that we have a branched chain, like the intact lipids 15 I just showed you before, and that we have lost, we have downregulation of the short chain lipids. And this downregulation could tentatively be explaining why we have lost some short chain unsaturated, monounsaturated compounds and gain branch chain short chains. So it means that at least part of the lipids we have in the membrane upon the pressure stress were transformed in situ, so within the membrane, and didn't necessarily imply that we have de novo synthesis for the whole set of lipids. So transformation at the level of the lipid substituents is clear under pressure, and so in contrast to what we saw for the intact lipid polar heads, which were not changing for Shewanella. So we can conclude that we have both for these different bacteria, the gram-positive, the Staphylococcus, and the gram-negative, different behavior in front of pressure. So the gram-pos had a complete change in polar heads with a development of ceramides. So clearly there was synthesis there. And in contrast, so we, for Shewanella, for the gram-negative organism, all the changes, all the changes in the membrane properties were associated with in situ, at least partly in situ, transformation of the existing membrane lipids. So there is a general problem with the lipid experiments in that we have, when we do those analyses, we are confronted to a very large abundance of unknown compounds. So yes, we might know they are cardiolipin or ceramides, but we don't know about the specifics of their structure. And there, in this large abundance of unknown, for families like ceramides, we might have as well transformation changes in the lipid substituents that are going in the same or different directions. So the general conclusion of that type of experiments that we need to try to figure out why those polar head changes. So there are a number of hypotheses, of course, and we will continue these experiments by doing longer growth experiments. And for the ocean worlds, we need to try, of course, conditions that are more adapted to their particularities, with changes in salinity, pH, and temperature, and these type of experiments will really lead to a better understanding of adaptation to these extreme conditions. So these poly-extreme conditions, high salinity, high pH or low pH, and as well as of course temperature, because of that is relatively cold. So these experiments are really interesting and provide a lot of surprise compared to what is estimated in the literature, as for example the increase in phosphatidylglycerol versus phosphatidylethanolamine, which was repeated in numerous publications, is not what we observed at all. So I thank the USC team, of course, and especially Stephanie Colonia in chemistry, who let us use some of her some of the mass spectrometer. And of course, we acknowledge that we're part of the Habitability of Hydrocarbon Worlds Astrobiology Institute, which is led by Rosalie Lopez and Michael Malaska. And we thank the whole team for their discussions when we meet. And of course, we thank Astrobiology for the funding and NfoLD for the discussions and interesting developments. Thank you so much.
Thank you, Fabien. And we'll go straight to Will's presentation now, and then we'll open up with questions and discussion.
All right. Let me get the screen back over here. Share full screen, hopefully. Can you see that okay?
Yeah.
Yes, it looks good.
Okay. Wonderful. I don't know if there's a sidebar with a camera, but on or off, doesn't matter to me. So thanks a lot. Yeah, I'm trying to absorb all the knowledge that I just received and understand how some of these experiments convey to what you might want to observe on another planet. And of course, there are many steps between there. The last thing that Fabien mentioned in the very last slide mentioned experiments that involve MS. That's actually a real term having to do with examining molecules and their internal structure by subsequently fragmenting them. Mm-hmm. And that is a very powerful technique. I think it has a huge future on these missions, but it also takes a lot of thought and a lot of time. And I'm fundamentally scared about missions that there's a clock ticking, and these amazing techniques that we have access to in the lab have to be very carefully chosen for their value in elucidating molecular structure when perhaps you only have a few hours of total analytical time. It's a totally different world to think about in the sense of doing science with your hands tied behind your back. So that's where I'm coming from to talk here, but I really appreciate this opportunity to talk about separation mass spec, which is one feature of how you would approach the analytical molecular analysis problem on a mission. It has some history and it has a great future, but I totally emphasize a little about it because all I can fit in this talk is really introductory concepts, and then I'll just invite discussion later. So you can see the three canonical ocean worlds talked about the most on the screen here. The application here is to ocean worlds, but the ideas certainly do have broader broader applicability. They do also apply to Mars. It doesn't have to pretend that it's an ocean world to be interesting to me. So a lot of what I'm going to talk about might also be of interest to the discussions coming out of MEPAG. So cast of collaborators, I just want to acknowledge up front my amazing group of colleagues and collaborators on various projects. There has been a significant development recently on this Emily project, which I will talk about later in the talk. That's in partnership with JPL, led by Peter Willis, and NASA Ames, led by Tony Rico, and a French consortium that's led by Cyril Zopa and team. And that's all going on. We're attempting to get that project moving forward, but we're working in parallel with the Titan mission. The DRAMS experiment, the Dragonfly mass spectrometer, is a big thing happening where I work at NASA Goddard. That is being led by Melissa Traynor, and then in parallel with that is really a broad group of scientific collaborations on other missions and projects, which include NfoLD teams. So there's a lot to do and pay attention to when you're thinking about taking experiments and applying them to future missions. And it takes a big team to carry out all of the interesting things, but also to decide what are the priority projects to focus on, because there's only so many hours in the day. And just to acknowledge that the Emily funding in particular is most recently coming from Matisse. So I'll try to get through these topics. The background on techniques. Let me see if I can. Okay, I'm just gonna see if I can remove the screen that's blocking a little bit on the right there, but I will move forward as fast as I can. The background on techniques has got to be really brief, but again, just to invite conversation later. Any questions on or offline are totally welcome. I'm not personally formally trained in separation mass spectrometry, so I'm reporting what I have accrued in discussions with others, but I'm coming up to speed on the lingo, and certainly seen quite a few of these experiments turn into flight projects so far in my career. So I'll talk a little bit about the concepts and the types that get talked about the most, and then I'll give my one kind of main example, the Emily separation mass spectrometer. I'll tell you what that stands for later. There's probably not time in this talk to go into depth on DRAMS itself, but of course questions are welcome. All right, let's see here. Whoops, let's go back up. Yes, I can use a keyboard. Okay, so just a little background. Maybe I'm preaching to the choir here. I know I am, but I like to restate basics. So, why do we prioritize looking for organics? Not just lipids. Lipids are awesome, and they probably form a center point of focus among all of the organic chemistry that we're searching for on other worlds. But if I just went through the logical basis for this, it is quite interesting to realize the assumptions that we're making about organic biosignatures. So, if an ocean world has life, it's mostly microbes, probably, and may not, I'm thinking that it's less likely we're going to come across a 12-legged squid as our first biosignature. So the microbes themselves may or may not be easy to see clearly. In other words, the manifest biosignature of a microbe in the center of your microscope, it's moving around, and people are actually thinking deeply about how we get that observation done. But microbes are presumably made of organics. They use organics and they produce organics, and those could distribute quite broadly. Any of those organics as a membrane in a cell or as part of metabolism could be a biosignature. And the fact that they could find their way intact, in other words, you would observe them as they originally appeared in the biosphere into accessible samples, is really the logical underpinning of why we would seek organic biosignatures and the entire chemical context in which they are found as valuable steps toward understanding if there's life on that world. Okay, so maybe that's all obvious to you folks, but it should be stated. So what we mean by this is it's involved, detecting and characterizing a range of organics in a mixture. And you want to do that as comprehensively as possible. So you want to know what organics are present. You want to know the various numerous biotic versus abiotic structures. There are hypotheses that we can test, and the criteria can be tested by measuring as broad a range of organic compounds and patterns as possible. So some of those hypotheses were captured very well in the Europa Lander study. I'm sure many of you have heard of or seen of that. It's basically some very nice graphics summarizing that I stole from the report in the lower left, summarizing the types of biosignatures that we would look for, and all of the molecules, the complex molecules, the molecular patterns, the isoprene units, the Lego principle stuff, bacterial archaeal cell walls, and essentially complexity generally, as realized in a set of molecules or even individual molecules that are potential biosignatures. That's all captured in like column 1, which is you just want to know what's there and you don't want to miss anything. So the driving requirement for a separation mass spectrometer is to give you the breadth of analytical sensitivity in all possible cases of what that environment might give you in a sample. That's a huge requirement, but I think it's absolutely important. In addition, we have some narrow and super powerful hypotheses like the chiral, enantiomeric excess. So enantiopure chiral compounds are found to be directly associated with terrestrial biology, and it's hypothesized that that's universal. So it'd be crazy to go and not try to make that measurement if you were to come across a population of chiral compounds that could be biosignatures. Okay, so that's another one. And of course, we all know about isotopic indicators. It also goes to fundamental physics about what life does and does not do metabolically, and uses light carbon, for example. So again, if you're looking for signatures like that, you may come across an organic molecule. On its own, it's a little challenging to measure and then interpret whether that one molecule that is observed to be fractionated is a sufficiently representative piece of biochemistry. It's really important to see pools of carbon and see whether the dispersion of isotopes between potentially biotic and abiotic compounds is represented in your total sample. So that just goes back to the first requirement, which is to see as many molecules as you can, ideally all the molecules that are there. At least I can dream. So these involve detecting and characterizing organics in a mixture, and that's really what a separation mass spectrometer is designed to do. It allows these types of analyses across the widest range of compounds, and potentially enabling one to directly distinguish biotic and abiotic signatures, of which we have hypothesized ones from our terrestrial studies, just as we have heard. Things like the distribution of lipid carbon chain numbers. Okay, it's enough on that. That's all the basics. So if I were to start now drawing cartoons, here we get into a little bit more of the basic idea behind SMS. I'm going to stop saying separation mass spectrometry, or I'm going to run out of time. So the idea is we do want to separate molecules in advance of their molecular analysis by a mass spectrometer. So in particular, what is being separated? Here, the definition of the technique is really analyte compounds that are extracted from a mixture into a fluid, which is a gas or a liquid. The main SMS power comes from its ability to render a complex mixture of molecules, these unknowns, in an unknown matrix over a wide concentration range and make it amenable to the precise molecular analysis by MS. The MS shines when you give it molecules in certain amounts in a certain order, and you don't overly complicate the mass spectrum. And so it stands as a simplification, but also another dimension of analysis, which is mentioned in another bullet here. So I'll just keep going down. It enables discrimination of isomers that I've mentioned in time, and those can be then quantified by MS. Again, if you have cleaned up / simplified the population of molecules that the MS has to deal with at one time, you stand a much greater chance at doing proper quantitation. And the front end itself provides something like retention time. It's basically another number, another axis of molecular characterization data to complement the molecular weight. So again, these are very general principles. not getting into examples, but it's interesting that we just saw those two plots on the previous slide showing like a Fischer-Tropsch type generation of a distribution of lipids, and then on the bottom lipid molecular weight distribution that is a fingerprint of a biogenic process. Okay, so as I trip over my words to describe these two plots, the idea of an SMS is that if you were to simply take all the molecules that are separated out in time on these chromatograms and pour them into the mass spectrometer all at once, you would have a very hard time discriminating what was an originally distribution of chain lengths in a bunch of alkanes or the fatty acid aliphatic chains versus those that are appearing over that same range of molecular weights due to fragmentation. So that's just one example where you would be confused if you didn't have the additional retention time data to work with to help identify the compounds in the MS. Here's the cartoons here, they're pretty self-explanatory. You basically have a pile of sample. Here it's snow because I'm thinking about ocean worlds that are icy on the top. So it's loaded into some kind of container, an oven or an extractor, and it's processed under controlled temperature and pressure conditions. It's really more than even just a condition. It's an entire preparatory program that you build into the sample processing. It's likely in the presence of reagents that are mixed with the sample and the matrix to aid in the release of different compounds. In the case of gas chromatography, on the top, the evolved gas is introduced to the GC. There's lots of details that are not shown here. And the compounds are separated into packets with different retention times in the column. So most people are aware of that. It's the general idea there. That column is many, tens of meters long. So these elutions can take tens of minutes depending on the flow rate, et cetera. So these are emitted in a time sequence, usually over tens of minutes into the MS. So you basically have an extended MS experiment, and every fraction of a second, you're getting a complete mass spectrum. So it is a pretty big data set, and it's extremely powerful data set. Similarly, in the bottom cartoon, the liquid chromatography or capillary electrophoresis are the two acronyms here. I'll try to define these as we go, and I have a list at the end. Here, processed and presumably concentrated liquid is injected into a capillary, and it's separated as liquid flows through. And the mechanism of separation is totally different than a GC, but the general idea is that you create a time sequence of analyte coming out and entering an MS. And the emitted liquid is normally sprayed into air. I mean, tiny droplets come out of a tiny tip. If you apply a high voltage between that tip and your mass spectrometer, you get electrospray ionization. And it's essentially the easiest / most powerful way of getting these ions into the MS from the liquid state. And these two techniques are quite complementary. Some things are, more volatile compounds that tend to want to get into the gas phase and are not as amenable to being dissolved in water or other liquid reagents. And other compounds, polar compounds, water soluble, are more amenable to being analyzed in a liquid protocol. So I will tell you the ideal combination of having both of these online and available for any given sample on an Ocean Worlds mission is kind of where you want to get to, if you can fit it. That's basically the love and the pain of my job every day, if you can fit it. So yeah, here's what my disclaimers here. These are not really puffs of gas. Those of you who actually know these techniques realize that my cartoons are just cartoons. I just thought they looked cute. And those are not really droplets of water. This is a continuous flow. The things that are actually bunching up are molecules with common, retention times or, elution properties.
Hey, Will, we
Yeah.
only have 8 minutes till the top of the hour. So I just
Okay.
wanted to let you know. And
All right. Yeah.
your enthusiasm for
Yeah.
separation got away with you.
Yeah, thank you. Okay, so I'm going to skip over some of this introductory stuff. It is my enthusiasm. I kind of just want to convey the general idea of the separation MS. All right, so this slide will just time step through. Thanks for the heads up, Heather. These are a sort of example applications and example implementations of some of these major separation MS methods. So GCMS on the left, most of you are familiar with the SAM experiment on Mars, uses a 6-column GC. And essentially, as I said, non-volatile polar analysis is possible with the derivatization and thermochemolysis. You can analyze fatty acids. So this is today's topic by transforming them into fatty acid methyl esters. GCMS of large lipids, however, is difficult. And so that is somewhat of a driver to wanting to combine GCMS and a liquid protocol. The two major liquid protocols that I mentioned, CEMS and LCMS, are themselves quite complementary. They use different techniques for separating compounds. LCMS is kind of the laboratory standard bearer for analysis of the widest range of heavy and complex molecules. However, CE has been demonstrated to more or less match the analytical capabilities, and in some cases get quite a bit lower in limit of detection. And so if you're looking for the range of very small to large, and I'm thinking like amino acids to peptides, in addition to being able to see kind of larger lipids, there are some distinct advantages to having the CE system, for which it is possible to analyze the wide range of molecules within one or a few runs. LC has an incredible amount of analytical power in the lab because of your ability to tune the mobile and stationary phases and actually get incredible powerful results and quantitation down to very low limits of detection for lipids and large biomolecules. But they tend to, this is my sort of personal awareness, which is not high, they tend to require more parameters to tune the experiment. And so because of that and the consumables, it has not moved as quickly on the TRL scales in terms of like, it's not on a mission yet. But that's obviously a motivation for future development. There's certainly been quite a lot of miniaturization. Okay. All right, so here I'll just more or less land on Emily, and I'll just give you the basic intro part of it. And if there are questions, I can show some of the examples and the data. So the idea is saying in a graph that what I had said before, which is that you want not to leave a molecule behind if you're not sure if it is important or not. To say no molecule left behind, really it means there is a phase space of potential molecular biosignatures and the contextual organic chemistry. And that means that you want a complementary set of techniques that allow you to see polar and non-polar compounds and volatile and non-volatile compounds. So that's more or less what's shown there. There's a lot more detail about the background of that motivation in a paper that I can forward to anyone who's interested. Our engineering concept for what this might look like on a, say, Europa or, Enceladus Orbilander is shown in the upper right. There's a lot of detail in there. It's just the idea of what all the components and boxes that are involved in having a dual multimodal, really, separation mass spectrometer. I'm sorry, is someone talking? Because if you need to cut me off, please go ahead. Yeah. So
I think you just have an open mic.
Oh, okay. Pardon me. So the fundamental design of Emily that realizes this kind of no molecule left behind approach is that we combine the capillary electrophoresis protocol through the oceans subsystem, which does, separation mass spectrometry and more. And you've probably heard from Peter Willis and his group about the developments there and the capabilities, combined with the GCMS, which we call GAPS, which is a multi-column GC and an evolved gas analyzer. And more or less between the two of those that are linked to a common ion trap mass spectrometer that has, well, it has MS and MS. We haven't really pushed it to MS to the N too much, although it's totally doable. We're more or less focused on trying to capture the complementary views of the molecular population by using a common mass spectrometer and really honing in on getting as precise molecule identification we can through that broad assay. Okay, I'm going to stop there because I do appreciate you need to have time for questions and whatnot in the open forum, and I can certainly add info and slides that are subsequent to this on some of the details and some example data, but we'll stop there.